In this video, we will be talking about restriction enzymes. Restriction enzymes are able to cleave DNA or cut DNA sequences at specific sites. They are either found in bacteria or used in biotechnology. Restriction enzymes in bacteria. So, in bacteria, they are used to restrict foreign DNA that enter the cell by destroying it. For example, if it were not for restriction enzymes, this virus would inject its viral DNA into the bacteria cell, and it will be able to replicate itself and form more viruses inside the bacteria cell and eventually break the cell. So restriction enzymes, these purple dots right here, bind to the DNA and cut it into smaller fragments so that it will no longer be able to replicate itself. So basically, the restriction enzymes are kind of a guard or a form of protection for the bacteria cell. The sequences that are usually recognized by restriction enzymes are 4 to 6 nucleotides long. For example, the restriction enzyme EcoR1 recognizes the sequence GAATTC, and its corresponding strand has the same sequence running in opposite direction. After the restriction enzyme passes over this molecule, it cuts it. This cut can either be a blunt cut or an uneven cut with sticky ends. In this case, it has a nucleotide strand that overhangs at the end, and this overhanging nucleotide is called a sticky end. These strands are complementary to each other, which enables them to pair together. Then DNA ligase passes over them and joins the strands and reforms the phosphodiester bond of DNA. As a result, it creates a molecule of recombinant DNA. Restriction enzymes in biotechnology. They are used to cut DNA into smaller strands in order to study differences and similarities in fragment length among individuals, and also scientists could use restriction enzymes to manipulate the molecule for research, as in gene cloning or DNA fingerprinting. So why are restriction enzymes so important? The reason is because any two fragments of DNA produced by the same restriction enzyme can be joined together. That's it.